So good evening, everyone. On behalf of Pediatric Hematology Oncology Chapter of Indian Academy of Pediatrics, I welcome all of you to this special webinar. The topic for this webinar is Mechanistic Insights into Anemia. And to give this talk, we have none other than Dr. Mohandas Narla, an Indian origin doctor who is now the president of American Society of Hematology. So he has got a very long CV, but I'll just briefly give some highlights. The most interesting thing is that sir actually did his chemical engineering in India. So he did his BTech from University of Madras, then MTech from IIT Kanpur, and then he did Doctor of Science in Chemical Engineering at Washington University. Subsequent to this, he was member of adjunct faculty laboratory of blood and vascular biology at the Rockefeller University, vice president for research at the New York Blood Center, and is the distinguished scientist at the New York Blood Center. He served at the post of vice president, then president-elect, and then the president of ASH in 2024. His research interests during the last 50 years have focused on developing detailed mechanistic insights into red cell function in health and disease. Current research efforts include studying red cell membrane biogenesis and erythropoiesis in health and diseases, including sickle cell disease, thalassemia, malaria, DBA, and myelodysplasia. Specifically, the research efforts include defining the contributions of reduced cellular deformability to increased red cell destruction in various human hemolytic disorders and the study of normal and disordered erythroid differentiation in various red cell disorders like DBA, sickle cell, thalassemia, MDS to define contributions of ineffective erythropoiesis to anemia. So we have all read your articles on various topics, hereditary spherocytosis, and many others. And we are really honored to have you here today to speak on this important topic of mechanistic insights into anemia. Welcome on behalf of Pediatric Hematology Oncology Chapter of Indian Academy of Pediatrics. And we are all eager to listen from you.
Thank you so very much. I really appreciate the invitation, and I'm sorry I can't be there in person, but I hope one of these days I can attend your meeting. Before I start, I want to say although my name is Mohandas Narla in America, it's really based from India, where I come from, Andhra Pradesh. I always write the first name last, so all my publications are under Narla Mohandas. So I just wanted to clarify that. Second is I'm quite very happy because my daughter happens to be a pediatric hematologist at Stanford. So it's a pleasure. I always enjoy working with pediatricians. A lot of my work I have done because of hemolytic anemias has been done with my colleagues who are pediatric hematologists in this country as well as in France. So it's really a pleasure and honor to be with you all this morning, or this evening for you. So as was mentioned earlier, but I've been started my PhD thesis in red cells from an engineering perspective about in early 70s. And I was so impressed with cells and cell biology that I stuck with it rather than doing engineering stuff. And I'll try to summarize for you what I have done over the last almost 50 years now. In terms of this morning's talk, I'm going to give you a broad overview of anemia rather than the specific diseases, but I'll illustrate some of the points I'm going to make with specific red cell disorders. Can I have the next slide, please? As you all know, anemia is a major global health problem affecting approximately 2 billion people or even maybe more. So there's really a serious public health issue. Anemia is a serious public health issue. And the causes of anemia are one, increased rate of destruction of circulating red cells in disorders that include red cell membrane disorders, sickle cell disease, immune hemolytic anemia, nutritional anemia, and malaria. The second cause is decreased production of red cells in the bone marrow due to ineffective erythropoiesis that include thalassemias, congenital erythroblastopenia, and hematologic malignancies such as myelodysplasia. I need to point out that although these are the two features of accounting for anemia, in some of the disorders, as I'll mention briefly, both of them can take place. Like in sickle cell disease, we have increased rate of destruction of circulating red cells, but also ineffective erythropoiesis. They are not exclusive, but in general, one of the two mechanisms is the dominant cause of anemia in human beings. Can I have the next slide, please? So what I'm going to do today is split my talk into two sections. In the first section, I'll talk about anemia due to increased destruction of red cells. These include, as I said earlier, red cell membrane disorders, sickle cell disease, thalassemia, autoimmune hemolytic anemia, and also in malaria, where there's increased destruction of red cells causing anemia. A distinguishing feature of non-nucleated human red cells is their ability to undergo extensive deformation during passage through the microvasculature for effective oxygen delivery to the tissues. Membrane cell surface alterations and decreased deformability can decrease the red cell lifespan. Normal red cell lifespan is about 120 days, and in this anemia, it can vary anywhere from 10 days to 90 days or 100 days. So, depending on the extent of decreased lifespan, the anemia is obviously more serious. So I'll show you some example how it could be in sickle cell disease. We have know a lot about structural organization of red cell membrane over the last 30, 40 years. And we have a pretty good idea of all the membrane components. Actually, in the red cell membrane, I've shown you a few of the major proteins in the red cell. But recent proteomic analysis has shown there are about 200 different membrane proteins in the red cell membrane. And we also know how this membrane is organized. And any mutations in any of these major membrane proteins results in red cell membrane disorders. And I'll illustrate that to you with some examples as I go along. Can I have the next slide, please? The red cell membrane disorders that I have been studying and my colleagues have been studying over the years are both structural defects, that is, the membrane structure is altered due to defects in some of the structural proteins of the red cell membrane, which I'll show you, and this results in hereditary spherocytosis and hereditary elliptocytosis. On the other hand, red cell makes a very big effort to maintain its volume around 90 femtoliters. And if there are defects in permeability defects due to change in the transport properties of the membrane, you can cause two types of diseases. One is called hereditary xerocytosis, and the other is hereditary hydrocytosis. In hereditary xerocytosis, the red cell water content goes down due to permeability defect, and the cells become dehydrated and have increased mean cell hemoglobin concentration. In contrast, in hereditary hydrocytosis, the red cells picks up water due to changes in sodium potassium permeabilities, and they swell and they become more stomatocytic. So these are the four red cell disorders I'm going to briefly describe in terms of molecular basis and our understanding of the root causes of these four disorders. Next slide, please. Hereditary spherocytosis is one of the most common red cell membrane disorders. It occurs all over globally, everywhere. And basically, its features is the fact that you see instead of being biconcave discoid red cells, you get cup shapes or what called stomatocytes, or also true spherocyte as shown on the right. So this is a very common, most common of the red cell membrane disorders. Can I have the next slide, please? It occurs in all racial and ethnic groups. It's common in Northern Europe, about 1 to 2,000, 1 to 5,000 prevalence, but maybe more common. And I'm sure it's a problem in India as well, but because it's not as extensively studied, we don't know exact prevalence in India. It's inherited dominantly in 66 to 75% of the cases, and non-dominant inheritance about 25% of the cases. I'm aware of at least 4 or 5 different cases of fatal cases reported hydrops fetalis due to very severe mutations in both parents. Anemia, jaundice, splenomegaly, and gallstones are a constant feature of hereditary spherocytosis. In fact, in some of the individuals, there's no diagnosis made till they are 30 or 40 or 50 years old because of gallstones. And then people do the testing and show that, in fact, they have mild forms of hereditary spherocytosis. Next slide, please. The in terms of classification, one thing I want to mention is in all these red cell membrane disorders is very phenotypically very variable. So the anemia can be mild, moderate, or severe depending on the hemoglobin level, circulating hemoglobin levels. Mild being 11 to 15, moderate 6 to 12, and severe being 6 to 8. And going along with the severity is the retic count, where in mild diseases it's 3 to 8%, whereas in severe it can be more than 10% reticulocytosis. And same thing with bilirubin levels increasing with increasing severity. And in terms of morphology, as I said earlier, in the mild form, there are very few spherocytes, so sometimes it's difficult to diagnose, but clearly you can see, if you look carefully, some spherocytes in the peripheral blood smear. Whereas in moderate, there are more spherocytic cells, and in severe, you can see spherocytes all over the blood smear. Can I have the next slide, please? We know a lot about the mutations responsible for hereditary spherocytosis. As I said earlier, the major structural component of the red cell is what's called spectrin-based membrane skeleton. And this membrane skeleton is linked to the lipid bilayer in which the membrane proteins are embedded. And the major protein is band3, which is the anion transporter in the red cell. And this is binds to a protein called ankyrin, which then binds to spectrin, linking the membrane skeleton to the lipid bilayer. When you have mutations in any of the proteins involved in these linkages, either band3 or RH or ankyrin or spectrin, you loosen the association with the membrane skeleton and the lipid bilayer. And so as the cells circulate and age, they actually lose more and more surface area, and then they become more and more spherocytic. So this loss of linkage is responsible for loss of surface area through the formation of vesicles and during circulation. And the extent of anemia is very much directly related to the extent of cell surface area as they become more and more spherocytic. Go to the next slide, please. In contrast, of loss of this vertical linkages and cohesion of the membrane, in case of hereditary elliptocytosis, the defects are in the membrane skeletal proteins itself. In membrane skeleton, as I said earlier, is a key component of regulating the stability of the red cell membrane. And this is basically driven by the so-called spectrin-based membrane skeleton, which consists of a heterodimer of alpha spectrin and beta spectrin. And this heterodimer of alpha and beta spectrin come together and form a tetramer. So by head-to-head association of these two proteins. And anytime this self-association state is not strong, that is, either due to mutations in alpha spectrin or beta spectrin, then you get elliptocytosis. Also, the spectrin is also forms a ternary complex with actin filaments and protein 4.1. And this junctional complex is also very important. And if you have mutations in spectrin or protein 4.1, you actually again weaken the strength of the membrane skeleton and the fragment as they go through circulation. So in hereditary spherocytosis, the linkage of the membrane to the skeleton that's responsible for spherocytosis, whereas the weakening of the interactions in this skeletal structure itself is responsible for hereditary elliptocytosis. Can I have the next slide, please? One thing for people like me working on red cell, the most important organ is the spleen. Travel through the spleen is really a risky business for both spherocytic and erythrocytic red cells. And that's why you get splenomegaly in all these red cell membrane disorders. And this is really very well documented in the sense that in case of spherocytosis, the more surface area loss, the more splenomegaly you get. And same thing in the case of hereditary elliptocytosis as well. Can I have the next slide, please? So just to summarize this part, in terms of hereditary elliptocytosis and spherocytosis, splenectomy results in a significant increase in hemoglobin levels and decreased reticulocyte counts in individuals with moderate to severe anemia. But I must point out that there's very good evidence lately that overwhelming bacterial infections and thrombosis are long-term risks following splenectomy, and this needs to be taken into consideration in managing these patients with these two membrane disorders. And due to increased awareness of late complications of total splenectomy, partial splenectomy is being used at some centers to decrease the severity of anemia, especially in children, while retaining residual splenic function. The challenge with that in long-term follow-up is the spleen will regrow back again as the children age. And so in some cases, you have to go and perform total splenectomy. So it's a choice between the balance and the risk of thrombosis and keeping the children going for a while till they develop all their immune functions. So it's a challenging thing, but I think something to keep in mind. But there's no doubt that splenectomy really ameliorates anemia and decreases severity of anemia. And the next slide, please. The second aspects of red cell membrane disorders, what I told you so far is structural, that is either loss of cohesion or loss of stability of the membrane. But as I said earlier, there's also red cell membrane permeability defects. Can I have the next slide? So as I said earlier, one of the things is the hereditary xerocytosis. This is autosomal dominantly inherited disorders. Anemia is mild to moderate. Target cells and vesicocytes and stomatocytes are seen on smear. And the important feature is increased mean cell hemoglobin concentration. Because the cell is losing water and there's the same amount of hemoglobin, the concentration of hemoglobin within the red cell increases markedly, say from 33 to 37 to 38 grams per deciliter. And because they lose cell water, basically, when you do osmotic fragility tests, they have decreased osmotic fragility, which is in contrast to serocytosis and elliptocytosis. We have increased osmotic fragility, which has been long used as a test for these disorders. And important point I want to make about hereditary xerocytosis, it is not that rare as people think, because a lot of diagnosis is missed because the anemia is very mild. And so the best way to do is really to keep an eye for it. And the reason to keep an eye for it is there is propensity for severe iron overload as the children age. And actually, I'm aware of a number of cases from colleagues in different parts of the world where actually the patients died due to cardiac iron overload with hereditary xerocytosis in the 30s and 40s. So it's something to keep in the back of the mind about red cell membrane disorders. And the treatment is supportive, and splenectomy, in contrast to hereditary spherocytosis and elliptocytosis, is contraindicated and in fact can be deleterious because you get thrombosis following splenectomy, and we don't understand why this is the case. Next slide, please. And about 10 years ago, we didn't know the mutation responsible for hereditary xerocytosis, but now we know it is most 95% of the cases are due to mutations in this protein called PASO1, which is a mechanically stress-activated channel that enables the red cells to pick up calcium and lose water as a consequence of that. And in fact, the people who discovered this mechanically stretched channel called PASO actually received the Nobel Prize a few years ago because it's not only important for red cells, but it's associated with a lot of other human pathologies as well. Can I have the next slide, please? In contrast to loss of cell water and dehydration, the second condition is hereditary hydrocytosis, where they actually pick up water and swell. And the molecular defect in number of cases is due to the mutations in RHAE, which is the protein mutation that's responsible for this. And RH is really important because, as you know, RH blood group system is really important for transfusion. And laboratory features are macrocytic red cells. And in contrast to xerocytosis, which have increased MCHC, they have decreased MCHC because they have picked up water. And there's, again, in contrast to the xerocytosis, there's increased osmotic fragility. And clinical splenectomy is not beneficial and is likely to be deleterious, and complications are hyperlipidemia, aplastic crisis, and thrombosis. So again, I really am sorry that I really don't know the prevalence of these diseases in India, but I'm pretty confident that it must be not that rare. And in fact, unless you look for them and look for subtle differences, they're not bad, except in severe cases where you have severe anemia, of course, you'll easily notice. But when there are mild anemias, it is important to notice them, as I said earlier, in some of these cases because of iron overload problems as well. Can I have the next slide, please? This one is a classic test for membrane disorders these days, at least in North America and Europe, where it's called osmotic gradient ectocytometry. And it's very diagnostic. You get very typical patterns of osmotic deformability profile for in elliptocytosis, spherocytosis, and xerocytosis, hydrocytosis. Unfortunately, this instrument is quite expensive and it's very hard to set up and use. But if reference centers, even in North America, there are only about 4 or 5 centers that perform this, and they send people usually send the blood to these sample centers, and they make the diagnosis based on the profile. And also, now they're available in Mayo Clinic and other places. Genetic testing to really understand the mutation that's responsible for these different types of red cell membrane disorders. And frankly, I think it's nice to know the mutation, but it doesn't really help. The most important thing is the clinical manifestation of extent of anemia in these different disorders. Can I have the next slide, please? So the other thing which I've been working on for many, many years is sickle cell anemia. And again, this is a single amino acid substitution of beta globin gene and resulting in making instead of hemoglobin A, they synthesize hemoglobin S. And basically, whereas hemoglobin A doesn't polymerize or is liquid state throughout, in spite of any oxygen tension, these sickle hemoglobin polymerizes upon deoxygenation. You can see a lot of sickle cell in the venous circulation. And obviously, the sickle feature is sickle red cells, chronic anemia, vaso-occlusion of capillaries, thrombosis, ischemia, multi-organ damage. And really, it is a disease of chronic hypoxia following birth. So it's really affects all organ systems in the body, and it can be very, very severe and hard to manage. And I'm happy to say that this year at the ASH meeting in San Diego in December, Dr. Deepti Jain from Nagpur, the pediatrician, I believe, is going to be the Ham Wasserman lecture talking about sickle cell anemia in India. And in fact, I didn't realize we wrote a textbook on sickle cell anemia, and we had a chapter there saying sickle cell anemia is quite mild in India, which absolutely is incorrect. And actually, when I said that to Dr. Jain a number of years ago in India, she said, Dr. Nala, you're wrong. We have all the manifestations that you see there because fetal hemoglobin is higher, but in fact, it's not uniformly distributed in all red cells. So I think the government of India is making big effort in trying to manage and document sickle cell anemia. I'm very happy about it because I think it has been underestimated, the severity of the disease as well as management of sickle cell anemia in India. And I'm very proud that Indian government is doing something about it. Can I have the next slide, please? Actually, I worked a lot on pathobiology of sickle red cells, and basically the increased cellular dehydration, which markedly increases the kinetics of sickle hemoglobin polymerization, leads to generation of rigid cells that cause significant disturbance in flow through the vasculature. Not only is the red cells less deformable, they're also adhering more strongly to vascular endothelial cells and other red cells. And these adhesion reactions really clog up the capillaries as well and leads to crisis. And besides red cell sickling, endothelial dysfunction is a key contributor to pathophysiology of sickle cell disease. So, as you see from here, there are multiple facets of pathobiology of sickle red cells that account for anemia as well as all the other complications, including painful crisis and organ damage. Can I have the next slide, please? Just to show you how variable the sickle cell disease is, this is some beautiful study by Charles Quinn recently at Cincinnati Children's Hospital, where he actually measured actual red cell lifespan in about 12 or 14 different sickle cell patients. And you can see in some of them, the red cells survival, they're all disappearing within 30 days, whereas in others is much more complicated, but now some of them live up to 1,800 days as well. So it's a very variable phenotype, and Israelis which completely don't understand the exact basis for this Although, as again I said, the extent of sickling is a major determinant of red cell lifespan in these patients. Can I have the next slide, please? And when you try to correlate this extent of lifespan with an anemia, you can see the best estimate of severity is absolute reticulocyte count, and not that relates quite nicely with the red cell lifespan. But other hemolytic markers really don't do a good job. For example, bilirubin levels and hematocrit levels and hemoglobin levels. So it's really much more complicated. But reticulocyte count is a very absolute reticulocyte count is a very good indicator of lifespan of sickle red cells in our patients. Can I have the next slide? So basically, the mechanistic basis of hemolytic anemia and sickle cell disease, one, clearly extravascular hemolysis and peripheral destruction of red cell is really leads to the responsible for decreased red cell lifespan in sickle cell disease. There is also good evidence for intravascular hemolysis, but it's been difficult to exactly determine how common it is or what the extent of intravascular hemolysis is, and that's something that needs to be studied more in detail in the future. And recently, there's been very good evidence, as all of you know, in Cooley's anemia or thalassemia major, ineffective erythropoiesis is one of the dominant reasons for anemia and thalassemia patients. And people didn't think ineffective erythropoiesis is really a feature of sickle cell disease. But now, in the last 5 to 6 years, there's very good evidence that ineffective erythropoiesis, due to increased apoptosis of erythroblasts in the bone marrow, due to sickling, is also contributes to anemia in sickle cell disease. And this is an area of active investigation in terms of exactly how much is this contribution of inactive erythropoiesis to anemia in sickle cell disease. Go to the next slide, please. Next slide, please.
Yes, sir, this was it.
That's fine. So, as I said, extent of extravascular hemolysis is highly variable from individual to individual. Higher fetal hemoglobin levels and co-inheritance of alpha thalassemia increases the circulatory lifespan of red cells. And treatment of hydroxyurea by increasing fetal hemoglobin levels improves the circulatory lifespan of red cells and decreases the extent of extravascular hemolysis. Phosphatidylserine exposure is one of the determinants of red cell lifespan, and it's difficult to quantitate the extent of intravascular hemolysis in sickle cell disease. And so actually, I'm really pleased that actually hydroxyurea is being increasingly used in India, and it's readily available and is not very inexpensive. Whereas this is a big problem in sub-Saharan Africa, where, as you all know, sickle cell anemia is also highly prevalent, but they have no access to hydroxyurea in these countries because of economic reasons. And the next slide, please. You can ask why it's important to make diagnosis of red cell membrane disorders or sickle cell. Basically, it's guide appropriate therapy. As I said, it's very variable from patient to patient, and it's really important to diagnose and quantitate the extent of anemia and explain it. And it also anticipate and prevent complications, for example, like iron overload due to ineffective erythropoiesis. And genetic counseling of family members is very important in terms of future pregnancies and diagnosis and expectant management of these children. As pediatricians, I'm sure you're very well aware of this. Can I have the next slide, please? So basically, I hope I convinced you that we know a lot about red cell membrane disorders, and splenectomy is used to manage red cell membrane disorders. And again, in the case of sickle cell disease, increasing hemoglobin levels with hydroxyurea can decrease anemia reasonably significantly and manage these patients better. But now I'm going to switch my gears and talk about the other aspect of anemia, which is anemia due to decreased production of red cells, which is also known as ineffective erythropoiesis. And ineffective erythropoiesis is first described very well with beta thalassemia major or Cooley's anemia, and since then has been found to be a feature of a number of other disorders, including sickle cell disease, and also is an important contributor to anemia in congenital erythroblastopenia disorders such as Diamond-Blackfan anemia. And now, more recently, there's a lot of interest in explaining anemia in people with myelodysplasia due to ineffective erythropoiesis in the bone marrow. Can I have the next slide, please? I just want to mention that human erythropoiesis is a really very complex process in the bone marrow. About 2.5 million reticulocytes are generated every second in the human bone marrow in normal individuals. And this production can be increased up to tenfold. You can make 25 million reticulocytes as a second if there's a need in severe anemia such as thalassemia or sickle cell disease and other red cell destruction disorders. And all of this erythropoiesis, making of red cells, start with obviously hematopoietic stem cells. And this undergoes a very complex process of cell differentiation to generate the mature reticulocyte. And for convenience, most of us in the field divide this whole complex process into three different stages. One we call erythroid progenitors, where the stem cell commits to differentiate along the erythroid lineage, and people have, by colony forming assays, have identified at least three different subsets of progenitors called large BFE, intermediate BFE, and colony forming unit erythroid. This you cannot distinguish based on morphology. You actually have to do colony forming assays to quantitate these different populations of erythroid progenitors. Whereas the second stage, where the progenitors start differentiation and making red cells, is called terminal erythroid differentiation. And here you can morphologically recognize them in the bone marrow, starting with the proerythroblast, which then differentiate into early basophilic, late basophilic, and polychromatic and orthochromatic erythroblasts. Based on the nuclear morphology and extent of hemoglobinization, we can morphologically distinguish these five stages in human bone marrow. At the last stage, the orthochromatic erythroblast, something very interesting happens where it doesn't undergo cell division in terms of generating two cells, but undergoes asymmetric cell division where it generates the enucleate reticulocyte and extruded nuclei called the pyrenocyte, which is rapidly phagocytized by the macrophages in the bone marrow, and you don't see them at all because they're all immediately eaten up by the macrophage. This then reticulocyte then matures first in the bone marrow and then in circulation to generate the mature biconcave discoid red cells in circulation. The next slide. So we spent last 10, 15 years in my lab trying to understand better this, or all the different stages of erythroid differentiation and generation of reticulocytes. And what we now know, when the stem cell or the CD34 cells, to be more precise, CD34 positive cell, will undergo 8 to 10 cell divisions every 8 to 10 hours to generate the proerythroblast. So it's really a complex process with at least minimum of eight different stages of erythroid progenitors, and we don't can identify all 8 of them, but I'll show you some data how we at least identify 4 distinct stages during erythroid progenitor development. The next slide. So basically, the only way to distinguish these progenitors is by what they call colony forming assays. So use methylcellulose cultures, put your cells, and then grow them in the presence of stem cell factor and erythropoietin, and come back 8 days to 14 days later and see the size of these colonies. And you can see the large BFEs are huge with multiple nodes and cells. And each one of these colonies has maybe 20 to 30,000 erythroblasts. Then what we call intermediate BFE, which has about 3,000 to 4,000 cells, and the small BFE, which has about 2 to 300 cells. So these are you can see there's a very large heterogeneity of these erythroid progenitors of the BFE stages, whereas the CFE, which is the last stage of erythroid progenitors, are much more uniform, and these colonies have between 100 to 200 cells. So what we have been trying to do is, since you can't do colony assays all the time to quantitate these things, is there something we can do using surface markers to distinguish these different developmental stages? Can I have the next slide? So using multiple markers like CD34, CD36, transferrin receptor, and CD105, we recently published where we can actually distinguish at least 4 distinct stages of erythroid progenitors. One is the so-called immature BFE, which is the one I showed you before, which is large colonies with tens of thousands of cells, or mature BFEs or immature CFEs. and mature CFUs. So now we are actually able, using flow cytometry and primary human bone marrow, to find out how many of these different development stages are present in the bone marrow. Can I have the next slide? And to show that there really are distinct development stages, nowadays everybody does what they call RNA sequence of these purified populations of cells. So what we did was, from primary bone marrow, we separated these different erythroid progenitor status, which we designate EP1, EP2, EP3, and EP4, and then do RNA sequence and proteomics to show that they are in fact distinct populations. And then it's a continuum going from EP1 to EP4 in the bone marrow. And then obviously the proerythroblast to different basophilic stage and poly and earth are all, we can separate them very distinctly, both based on gene expression as well as proteomic studies. Can I have the next slide? And this is the proteomic data. Again, you can see as you go from EP1 to EP4, some of the proteins become strongly expressed only in EP4, start very low in EP1, which is green. And the other proteins are highly expressed in EP1, indicated by red bars. And then it goes down as they develop into EP4. So we are very confident that these are really distinct stages during erythroid development. And the reason is important is because, as I'll show you, in some of these red cell hemolytic anemias, the blockage in the terminal in erythroid differentiation could occur at this progenitor stage itself. Can I have the next slide? So the heterogeneity of progenitor population appears to be related to the number of cell divisions of CD34 cells and surface markers CD34, CD36, CD71, and CD105 enable the isolation of 4 distinct subpopulations of erythroid progenitors. Gene expression, epigenetic, metabolomic, and proteomic studies of these purified progenitor populations have been performed to obtain insights into human erythroid progenitor biology. Can I have the next slide, please? That is the progenitor stage. That's the first stage I talked about. That is stem cells becoming progenitors, and then the progenitors generate proerythroblasts, which then subsequently divide, undergo 4 to 5 mitosis to generate the reticulocyte. So here, in contrast to progenitors, where I told you there are 8 to 10 cell divisions, here we have 4 to 5 cell divisions. And again, here the cell cycle, the cells divide every 20 to 24 hours, not every 8 to 10 hours. So really, they're very different biology between the erythroid progenitors and terminally differentiating erythroblasts. And I also said earlier, you can, in the bone marrow smears or you can really distinguish these things morphologically. You don't need surface markers. But as you can imagine, if you want to count 1,000 cells of this to figure out what stage is a problem, is a big monumental task. Can I have the next slide, please? So again, a number of years ago, we developed surface markers where we can clearly get all these 5 distinct stages of terminally differentiating erythroblast, from proerythroblast to all the way up to orthochromatic erythroblast. And we hear 2 different other markers called alpha-4 integrin, which is a self-adhesion molecule expressed on erythroblast, and band3, which I talked to you earlier about, which is the anion transporters. And based on their expression levels, we can sort all these 5 different stages. Can I have the next slide? One thing I want to bring to your attention is we do all these studies initially in vitro cultures of stem cells so that we can generate large amounts of them. But I always remind my students and my colleagues, we always need to be sure when we do these markers, they're actually useful not only for cultured cells, but actually primary cells from the bone marrow. And here I'm showing you that using these markers, that alpha-4 integrin and band3, we can use the same markers to get very pure populations of terminally differentiating erythroblasts at distinct stages of maturation. The only difference you'll see at the proerythroblast stage in early baso, where in cultures you generate a lot of what they call stress granules, which are not present in primary bone marrow. And that's because in the cultures we are stressing them a lot with very high quantities of growth factors like stem cell factor and EPO. But at the same time, having said that, we actually done all the quantitation of these things by RNA-seq and proteomics, and really don't see much difference between in vitro and primary bone marrow cells. Kind of the next slide. So, what is the use of this? As I said earlier, it'll be important to quantitate these different stages in primary bone marrow. And here I'm showing you data from about 15 or 20 primary human bone marrows. And you can clearly see that we get very nice populations, distinct populations of different stages of development. And more importantly, for every proerythroblast, we have 2 early basal, 4 late basal, 8 poly, and 16 orthochromatic erythroblasts, which what you would expect, doubling every time the cell divides if there's no loss of cells or apoptosis at these stages. So in normal, you don't have any apoptosis during differentiation, so you can get doubling every time the cell differentiates. So now we can use this now then to say in ineffective erythropoiesis, where is the blockage during erythroid differentiation in the bone marrow? Can I have the next slide? Also recently, because we had to do 2 different assays, 2 different markers, one for erythroid progenitors and one for terminally differentiating erythroblasts, we recently showed that we can now use, unfortunately, 10 color flow cytometry, which is reasonably complicated. But in fact, using this 10 color flow cytometry, we're able to distinguish all 4 stages of erythroid progenitors from EP1 to EP4, as well as the terminal differentiating erythroblasts from proerythroblast to orthochromatic erythroblasts. So if the facilities are available, we can in fact define stage-specific abnormalities in different causes of ineffective erythropoiesis in different red cell disorders. Kind of the next slide. So basically, I'm now very briefly going to talk to you how we are using this methodology to study ineffective erythropoiesis in thalassemia and sickle cell disease and MDS during terminal differentiation, and also ineffective generation of erythroid progenitors in Diamond-Blackfan anemia and MDS. Kind of the next slide. So as I said earlier, it's been known for a long time that ineffective erythropoiesis during terminal erythroid differentiation is cause of beta thalassemia major, because the red cells in transition from polychromatic to orthochromatic erythroblasts undergo apoptosis because of accumulation of excess alpha globin chains in the erythroblasts. So you see a big decrease in the transition from orthochromatic to polychromatic erythroblasts in this bone marrow aspirate from thalassemia major, Cooley's anemia patients. Can I have the next slide? So recently we used this method then to study what happens when you do gene therapy. This is done work with Michelle Sadland, Memorial Sloan Kettering. So we had a bone marrow before gene therapy, and you can see again there's a big blockage at the late stage of terminal erythroid differentiation. But following treatment with gene therapy, you can see we now start seeing more and more orthochromatic erythroblasts. So really, it's a very useful methodology to see how effective the treatment has been to rescue ineffective erythropoiesis, and obviously the hemoglobin levels go up following that. Can I have the next slide? Also, as I said, thalassemia and ineffective erythropoiesis has been long well documented, but there's been some controversy about the fact that is this also a feature of sickle cell disease. And here I'm showing you a bone marrow from a patient with sickle cell disease, and you can see in controls, every time the cell divides, you get 2 cells for every 1 cell. But in the case of the sickle cell bone marrow, you can see between polychromatic to orthochromatic, just like in beta thalassemia, there's a big drop off. Instead of being doubling, there's only increased 1/4. So that means you're losing cells between polychromatic and orthochromatic erythroblasts. And now we have validated this in a number of other sickle cell patients. And this is all due to hemoglobin polymerization at this stage of erythroid differentiation, because there's enough hemoglobin concentration that, in fact, you can see sickle fibers in erythroblasts at this stage, at the late stages of development. So I think we need to be aware that not only increased destruction, but also ineffective erythropoiesis can causes anemia in these patients. And in fact, if you increase fetal hemoglobin, you increase not only the red cell lifespan, but also decrease ineffective erythropoiesis in sickle cell disease patients. Can I have the next slide, please? Again, we are using this methodology to study other disorders with ineffective erythropoiesis. And I've been interested for a long time in Diamond-Blackfan anemia, which is what's called ribosomopathy. It's due to mutations in various ribosomal proteins. Now, I think mutations in 20 or 22 different ribosomal proteins have been found in association with Diamond-Blackfan anemia. The most common, about 20 to 25% of DBA patients have mutations in the originally identified ribosomal protein RPS19. And also the second most common is RPL5. And in fact, you can see that. Can I go back, please? Sorry. You can see that, in fact, we get distinct patterns of erythroid progenitor defects in RPS19 and RPL5 mutations. In RPS19 is little normal except the transition from EP3 to EP4, whereas in RPL5 you get problems even in the transition in the earlier stages of terminal thing. So I'm pretty confident we are going to be able to use this methodology to more precisely understand what specific stage is the defect in erythroid progenitors. And erythroid progenitor defect is a dominant cause of anemia in DBA patients. Can I have the next slide, please? And now, more recently, I got interested in myelodysplastic syndromes, which is not common in pediatric patients. Obviously, it's a disease of older people. And again, we find that the ineffective erythropoiesis is a feature of both terminal erythroid differentiation and in erythroid progenitor biology in MDS. And we are pursuing this further. And I'm doing this work in conjunction with my good friend Azra Raja from Colombia, who has a big MDS clinic, and we have studied about 200 bone marrows from MDS patients to date. Can I have next slide, please? So basically, in summarizing, based on distinct cell surface markers and metabolic transport expression of human erythroid, we are able to isolate at least 10 distinct stages of human erythroid cells at different stages of development. We are currently performing genomic metabolomic studies on these different subsets of erythroid cells to understand more detail the biology of these cells. And defects in erythroid progenitor biology is a feature of disordered erythropoiesis of various human disorders, including thalassemia, sickle cell disease, Diamond-Blackfan anemia, and myelodysplasia. We anticipate the methodologies we develop is likely to contribute to better mechanistic understanding of both normal and disordered human erythropoiesis. Can I have the next slide, please? So I talked to you a lot of different things. I hope I made a few of the interesting points about how we really know a lot more about mechanistic basis of anemia in humans, but we still have a long way to go to really get all the detailed understanding. And obviously, I didn't do it. Most of this work was done by my colleagues, including the person who developed all the methodologies for flow cytometry was Hongxia Yang, who was my colleague at the New York Blood Center, and a very close collaboration with my former postdoc, Lionel Blanc, who is now in Feinstein Institute in Long Island. And I work very closely with people around the world, and at Yale University with Pat Gallagher and Vince Schultz, we did all the genomics. My daughter, Anu, who is a pediatric hematologist and works on DBA, and Michelle Sadlan, I told you, with gene therapy, and Azra and Abdullah for MDS. And I started my career in hematology in Paris in 1973, and I've been working since then with my colleagues, Lydie DaCosta and Emily Fleur Gauthier. And most of the metabolomic studies are done with Naomi Taylor and Sandrine Kinnett in Montpellier. So I'm been very fortunate in my life that I've met so many wonderful clinicians. And I tell people like me who are PhDs who are doing this kind of research, the only way to be successful in hematology research is to really work with fantastic clinical scientists, clinicians, and clinical scientists to kind of help you understand what you're doing in the lab has relevance to clinical diseases. So I've been very fortunate in that regard. So thank you very much, and I'll be happy to answer any questions.
Thank you, sir, for giving a overview regarding your research for the last 50 years and how it has translated the practice in hematology. And I would request the delegates to drop their questions in the chat box. While we wait for the questions, like I would put one question, sir, in Diamond Blackburn anemia, like
Right.
how steroid work and how it gets better with age in some population.
Very good point, actually. As you know, the only therapy right now we have is steroids. Like about 50% of these children respond very well, to steroids, and they increase the hemoglobin and their transfusion requirements go down dramatically, stuff like that. And there's been always some inclination that, in fact, the steroids act at the progenitor stages. And in fact, we recently, with Lionel Blum, my colleague, we had a paper in Journal of Clinical Investigation that steroids actually act at the late stage of erythroid progenitors in the E3 to E4 stage. Huh? So if you have E3 and E4, in DBA, you get up to the E3 to E4 stage in the bone marrow, then the steroids can increase the production of red cells. They undergo terminal differentiation. In the absence of steroids, they don't undergo as much differentiation as with steroids. So that is reasonable account, but we still don't know why some people are not responding. That's the whole idea of trying to define exactly what stage the defect is. So maybe that'll give us, and maybe we can develop other agents which will stimulate the development of earlier stages of erythroid differentiation and stuff like that. So it's a very open, very important question. We don't have all the answers for that, but I'm pretty confident we know that steroids do act at very late stages of erythroid progenitors. It has been speculated many years, but now we have actual evidence to show that is indeed the case. Yeah.
Thank you, sir. We have one question from Dr. Siaram, like
Right.
when to go for splenectomy in hereditary spherocytosis.
Actually, it's the only thing, as I said earlier, it's very curative. Even people with severe anemia, you can manage without any transfusions for a long, long time. But I think one thing you need to be careful is, as I said, the thrombosis seems to be a problem as a function over time. So you just want to monitor and make sure aware of the patients to come and see you when there's something is changing or something is different. And my colleague Jules Chernier in Paris, who I worked for the last 50 years, was the first one to pioneer partial splenectomy because he was worried about children when they're very young. Their immunological system is not fully developed, and spleen does have immunologic functions. So he thought you should do partial splenectomy. And clearly, when you take about, we had a couple of papers in Blood on that, where you can show you can get at least with removing 90% of the spleen, you can really significantly increase the hemoglobin levels. But as you would expect, over time they did regrow back. So we have about a uh, 70, 80-year follow-up of these partial splenectomy patients, and a number of them had to be total splenectomy, where they started again, becoming a lot of splenomegaly and stuff like that. So the other thing I forgot to mention is that, in fact, other thing with spherocytosis we have to be very careful about is parvovirus infections, because they can really become, as you can imagine, as a pediatrician, that very severe anemia. And again, the idea is to tell the parents that if something is changing, they become lethargic or something, make sure that they immediately brought in and transfused, because it is a transient erythroblastopenia. But nevertheless, since this red cell production is increased maybe 5-10 fold in the patients with anemia, if you don't make red cells for a week, then you're in big trouble. Yeah.
Thank you, sir. A few more questions, like how
Oh,
do
sure. I know, all the time. I'm not in a hurry. So.
like how do hereditary xerocytosis and hydrocytosis present, and how do we medically manage?
Yeah, basically, the thing is, the present is hereditary osmotic fragility is a classic test used for spherocytosis for decades now, or maybe even more than that. And basically, both spherocytosis and hydrocytosis, they have increased osmotic fragility. So if you just do osmotic fragility, it's very easy to diagnose the thing. Only difference is in hereditary hydrocytosis, the MCV is 120, because they're picking up water and becoming bigger. Volume goes up dramatically. So by looking increased osmotic fragility and looking at the MCV, you can clearly distinguish between spherocytosis and hydrocytosis. In the cases of xerocytosis, as I said, the osmotic fragility is profoundly decreased because they can take a lot more water before they can, because they're dehydrated, they take a lot more water before they swell and lyse. So osmotic fragility test is not done mostly in the Western countries because it's expensive, it's time consuming, but it's a very simple effective test in my judgment to diagnose membrane disorders for sure. Yeah. And in terms of management, really, there's no drugs, right now. But I'll tell you one thing, one story about it. Basically, splenectomy, HS is very curative. You know, knowing what the complications are. And I said both for membrane permeability defects, xerocytosis and hydrocytosis, please do not splenectomize these patients. Because there's numerous cases of life people dying because of thrombosis. And unfortunately, after 50 years, I can't tell you why. Why they're thrombotic in these two conditions. When you take out the spleen, we have no clue. Something people have to really do a lot more studies to do that. And recently, there's a paper, which I was part of, where there's a new drug that is used to activate pyruvate kinase called miltipat or something like that. It's been phase 3 clinical trials on that. It was developed to manage pyruvate kinase deficiency. These people with pyruvate kinase deficiency with this drug really do a lot better after treatment. And now there's some evidence. Again, this is the pyruvate kinase. The story is very clear cut. There's a nice paper in New England Journal of Medicine about managing PK with this drug. Now, some people are trying think it may also be useful for sickle cell disease and hereditary spherocytosis, but the evidence is not is coming, but not there as strongly as PK. But it is promising, definitely promising drug thing, and we have to wait and see with well done, well designed clinical trials if it is really as good as its initial. Because most of the time the drugs work very well in initial studies, but then when you do large scale studies, they're not as effective. And also the big problem with that is the cost. The cost of these drugs is really, any new drug, as you very well know, is very, very expensive. And since it's easy to manage, you don't want to put somebody on a drug for a lifetime, which is very expensive. Yeah.
Thank you, sir. Next question is like, are there any membrane disorders which can manifest in neonatal age with severe anemia and getting transfusion dependent since then?
Oh, great question. Great question. Basically, I'll tell you, this hereditary elliptocytosis, is a big problem with neonatals thing. Okay, because what happens is they're very anemic at birth. Actually, in even people with a non-hemolytic HE, at birth they're very anemic. Their hemoglobins are 7 and 8 and all, but as the age, by the time they're 1 or 2 years old, their anemia goes away. And actually, my friend Bill Menzel was a great pediatric hematologist at UC San Francisco, and I had a paper many years ago. That's because what happens is, as you know, at birth, there's a lot of fetal hemoglobin, and then switches off as the age. By the time a year old, they come down to baseline. And what happens is fetal hemoglobin doesn't bind 2,3-DPG. Only hemoglobin A binds 2,3-DPG. And 2,3-DPG, unfortunately, has a destabilizing effect on the membrane. So the red cells on the neonates are much more mechanically fragile than after the switch of the fetal hemoglobin. So we studied a number of patients with hereditary erythrocytosis in neonates and as the age, and we can show very clearly, there's a paper in JCI where we actually showed that as the fetal hemoglobin goes down and hemoglobin A comes up, the anemia gets better. So it's really something to keep in mind, especially since this is hereditary condition. The parents are elliptocytosis and they have a newborn child. You want to be sure that you're aware that they're very anemic, but follow them. You don't need to do anything to follow them. If the hemoglobin keeps going up, you know that they're only heterozygous, and they won't develop severe anemia. Yeah.
Thank you, sir. And like, at what age do we prefer to start steroid in diamond blackfan anemia?
Actually, the reality is, at least I'm not very careful. As you introduced me very well, I'm an engineer and not a hematologist. But I became a hematologist by accident. Okay, so really, that's the. But I can tell you with some confidence because I've been working on this for a very long time. That basically people start them around a year old or something like that. Once the anemia becomes significant and stuff like that. Clearly, you know you can manage them by transfusions, but really that's not easy. I mean, depending on the blood supply needs, and stuff like that. But again, you have to be very careful because you're putting on steroids for a long time, right? I mean, if you stop it, the red cells are not made; they become anemic again. So it's really, you need to be very careful, and also dosing them determining the dose that they can maintain reasonable hemoglobin and cut down to the level where it is optimal to keep it going and stuff like that. Absolutely, because of all the steroid toxicities of long-term use. Yeah, yeah.
The next question from Dr. Neeta is, in spherocytosis, the
Right.
transfusion requirement decreases with age from infancy till the child is 5 to 6 years.
Right.
Why does it happen? Like, is it just related to the growth velocity which slows down as the child ages, or any other
Yeah,
mechanism?
that's like most likely explanation for sure. Yeah, because as you know, there's enormous increase in red cell mass as the children are growing. Their body weight goes up and stuff like that. And also we really, as I told you, the spleen is the dominant, the major, really, without a doubt, of destruction of spherocytic red cells. There's no doubt. We actually did some work with my colleague in Paris and Institute Pasteur, where we use perfused human spleens perfused with red cells to see how they go through the spleen and stuff like that. And where we showed in a paper in Blood that if red cell loses 16% of its surface area, the red cells are trapped in the spleen within the first or second passage through the spleen. So really, that's very clear cut. That is a filtering function of the spleen. Although you have to be careful because it not only filters, but there's a lot of macrophages in the spleen, which can phagocytose abnormal red cells as well. But I think the change in splenic function and, as you said, the amount of red cells you need to produce for stable values could both play a role in age-related clinical sequelae of spherocytosis. Yeah.
Another brief question. In PRCA, pure red cell aplasia,
Right.
GATA1 mutation, does this mutation have similar steroid responsiveness to the RPS mutation?
Actually, it's a controversial subject. There's recently a review article in Blood about a year back or something from my former student and now Professor Lady DaCosta and I in Blood, where we talk about these things. Clearly, GATA1 is a critical transcription factor for erythropoiesis. Without GATA1, you can't make anything, any red cell protein, whether it's globin or band 3 or any of the proteins. So clearly, GATA1 is critical for red cell biology. But there is controversy because there is some evidence that is ribosomal proteins, and some of them you have decreased expression of GATA1. But clearly, GATA1 is not the only thing that is responsible for defective erythropoiesis in DBA. It's more of a translational defect, because as you know, ribosomes are critical for protein translation, and now there's more and more. So I would refer, if you don't mind, to this article in Blood, review article in Blood. It really gives a very good summary of our current stage of understanding of DBA. And if somebody in knee can't have access, they send me an email. I can send the PDF of that. Yeah.
Thank you, sir. Like, just two more questions. Like,
No, no, no problem.
rare enzymopathies like pyruvate kinase, GPI deficiency, would
Good.
splenectomy be beneficial? Pyruvate
Uh,
kinase.
that I really don't know the answer, so I don't want to give you misinformation. I'm not aware of that.
And then can ineffective erythropoiesis affect the retic count as well, leading to low retic count? Traditionally, we are taught that thalassemia associated with increased retic count versus iron deficiency is having low retic count.
Right. Clearly, ineffective erythropoiesis can affect reticulocyte count because you're not generating, they're all apoptosis before they enucleate because they're apoptosing at the orthochromatic erythroblast stage. So if I reduce orthochromatic erythroblasts, they can generate as many reticulocytes as you would normally do. But again, my problem with this kind of in this subject is we really don't have good clinical data on large number of patients. You really need, to do that. And my hope, frankly, between you and me, that India, we have so many interesting red cell disorders. I think we can be able to really do a very good job of clinical investigation of these kinds of red cell disorders in India more than they can do in America or anywhere else. So really, I would strongly encourage the pediatric hematologist in India to really get into clinical, not fancy molecular biology and all, but good clinical observations. Because all these things I think is important in my judgment. I keep telling people all the time, having a longitudinal follow-up of patients, that is from birth till they become 14 or 15, whatever the time frame is, how the hematology is changing, the parameters are changing, and retic counts and hemoglobin levels and all, longitudinally in number, not two one. I can always give you case reports, that doesn't really do much. I tell you something, but you can't make generalizations about a disease based on a few case reports. So I would strongly encourage you and your colleagues to really keep that in mind and do very good clinical investigation about what the natural history of these different hemolytic anemias are in India. Yeah.
Okay. Thank you, sir. Thank you for your valuable suggestions. Definitely, we'll take it up. And I think, again, thank you once again for taking your time in answering all the questions in the chat box. And on the behalf of the Indian pediatric hematology oncology community, we would like to thank you for your time, valuable time, and your talk. And we would like to have you in person in one of our national conferences soon.
No, I would be delighted. Actually, I'll tell you one of my regrets, in my 50-year career, really, this is from my heart, I'm telling you this. I'd worked with people in France, England, Australia, Japan, everywhere, okay? In Thailand, on alpha thalassemia, I did a lot of work in thalassemia. I never had an opportunity to work in India, And at this stage, I don't know how much I can do, but I really would like to get more connections, with pediatric hematology, because I think there's so much to learn. And I really think you guys are so smart and you have so many patients too, which is important in this business, to really understand more deeply about these different disorders. So I'll be very happy to connect with you people, and really, I'm not at my stage, I can't do much, but I can at least encourage you and provide you uh, moral support to do very good clinical investigation. Yeah.
Thank you, sir. That would be wonderful.
Thank you very much, and have a good day. And it's getting late for you, so I'll leave you alone now. And thank you so much. Thank you.
Thank you, sir.
Bye bye. Bye bye.
So with this, we'll close the session.